Most cells in the body undergo mitosis as a means of reproduction. These cells are responsible for most bodily functions, excluding sexual reproduction. The only cells in the body that use meiosis are gametes, or reproductive cells. Common cells use mitosis because all they need to do is replicate themselves. There's no need for genetic diversity in normal cells. As a matter of fact, that would be a bad thing. Reproductive cells must use meiosis to create genetic diversity between themselves. Diversity comes with an evolutionary advantage. It creates a wider range of traits which are useful under different conditions. Starting off with mitosis. This is interphase. In this stage, the cell remains for the longest period of time. When it goes through interphase, it has a first gap, which is when it grows and makes proteins. Then it goes on to synthesis, where it copies its DNA. Finally, it goes to gap 2, where it scans all of the DNA and checks for any errors. The first stage of mitosis is prophase. Chromatin is visible and the cell enters a growth period, which includes DNA replication, supercoiled DNA, which strengthens the cell, and lastly, the nuclear envelope will dissolve and mitotic spindle tubes will latch onto chromosomes. The second stage of mitosis is metaphase. Metaphase is when all the chromosomes start to line up along the equator of the cell. The chromatids and spindle fibers are attached and the cells start separating all genetic material. Anaphase. This is when chromatids go to the opposite poles of the cell and they are guided there by the spindle fibers. Each end of the cell will get a complete set of DNA. Telophase. There will be visible cleavage in the cell, which helps identify that there are two distinct daughter cells. This completes round 1 of the cell cycle, and cytokinesis is complete. Moving on to meiosis, which is how reproductive cells reproduce. This is interphase. This stage of meiosis is essentially the same as the interphase in mitosis. The cell remains in this stage of meiosis for the longest period of time. It goes through the first gap, protein production, then synthesis, where it makes DNA, and finally it goes through its second gap, where it scans the newly formed DNA for any potential errors. This is prophase 1. This is the first and one of the most important stages of meiosis, where homologous chromosomes pair up and exchange genetic material. During this phase, the chromosomes condense and become visible under a microscope, and each chromosome aligns with its homologous partner to form structures called tetrads. A key event here is crossing over, in which non-sister chromatids exchange segments of DNA, creating new combinations of genes and increasing genetic diversity. The nuclear envelope begins to break down, and the spindle fibers start to form, preparing the cell for the next steps of division. This stage sets the foundation for the unique genetic variation seen in gametes and ultimately offspring. Fish is metaphase 1, the stage of cell division where the chromosomes line up along the center of the cell, called the metaphase plate. In meiosis 1, homologous chromosome pairs, or tetrads, align side by side, while in mitosis 2, individual chromosomes line up similar to mitosis. Spindle fibers attach to the centromeres of each chromosome, ensuring that when the cell divides, each new cell will receive the correct set of chromosomes. This precise alignment is crucial to accurate cell separation and helps prevent genetic errors in the resulting gametes. Anaphase 1. Homologous chromosome pairs are pulled apart to opposite poles of the cell, reducing the chromosome number in half. Sister chromatids remain attached. Telophase 1. Chromosomes reach the poles. The nuclear envelope may reform, and the cell prepares to divide into two daughter cells. Cytokinesis. The cytoplasm divides, physically separating the cell into two haploid daughter cells, each with half the original chromosome number. Prophase 2. Chromosomes condense again. Spindle fibers form and the nuclear envelope breaks down in each of the two haploid cells.
Because I'm still trying.
Metaphase 2. Chromosomes line up individually along the metaphase plate, with spindle fibers attached to each chromatid. Anaphase 2. Sister chromatids are finally separated and pulled to opposite poles of the cell. Telophase 2. Chromatids reach the poles, nuclear envelopes reform, and cytokinesis follows, producing 4 genetically unique haploid gametes for sperm cells, but only 1 for oogonia. The rest of the oogonia are reabsorbed. This completes the complete meiosis cycle in the complete reproductive cells and makes more of them.